Everything you think you know about the moment of death is about 30 seconds late. The flatline isn't the brain going quiet, it's the aftermath of something far stranger that already happened. In the very first seconds after a heart stops, the brain surges. Gamma waves, the kind tied to awareness and conscious perception, light up at levels that can exceed the waking brain. Then comes the wave. Then comes the chemistry. Then, on timescales you can barely imagine, the atoms disperse into the planet and eventually the cosmos. Science has mapped all of it except one thing, sitting right there at the beginning. Settle in, subscribe if you haven't, and stay to the end, because the one thing science still can't answer is the whole reason we're here tonight. Part 1. The moment science thought was silent. For most of the 20th century, the standard medical assumption was simple. When the heart stops, the brain stops. Cardiac arrest meant immediate cerebral shutdown. Within a handful of seconds, blood pressure to the brain drops to zero. Without perfusion, neurons cannot maintain their membrane potentials. Without membrane potentials, electrical signaling fails. So, the brain, the assumption went, simply switches off, like a lamp when the cord is yanked from the wall. There were good clinical reasons for this picture to dominate. It was simple, it was useful. It made decisions about resuscitation and time of death tractable. And for most of medical history, it was supported by the fact that we did not have the tools to look closely at what was happening inside the skull during the actual moment of dying. Patients in cardiac arrest are not usually wired up to high-resolution EEG systems. Animal studies, when they were done, often did not focus on the precise sub-minute window between perfusion loss and full electrical silence. So the picture of immediate shutdown was rarely challenged because it was rarely tested in the right way. That changed slowly, and then quickly, in the last 15 years or so. EEG monitoring became more common in intensive care units. Animal models, particularly in rats and pigs, started to look specifically at what happens in the brain in the seconds immediately following cardiac arrest. And what those studies found was not what anyone expected. The brain does not fade, not at first. In the first 10 to 30 seconds after circulation stops, there is a transient surge of organized electrical activity, specifically gamma band activity. Gamma waves in awake healthy brains are high-frequency oscillations, typically between 30 and 150 cycles per second, that have been associated with conscious perception, cognitive integration, and the kind of binding processes that link different brain regions into unified experience. They are, broadly speaking, one of the neural signatures of being awake and aware. And in a dying brain, in the first window after the heart stops, gamma activity surges. Not just appears, surges. By some measures, the synchronized coherence of gamma waves in the dying brain exceeds the levels recorded in the same brain when it was awake and engaged. There is also something called cross-frequency coupling, where the gamma waves interact with slower theta and alpha rhythms, and that coupling, which is associated with memory recall and complex cognitive operations, also intensifies. The dying brain, in other words, briefly looks more coordinated, more integrated, more functionally organized than the waking brain it just stopped being. This is genuinely strange. From a pure energy standpoint, it should not happen. Neural activity costs energy. Synchronization costs energy. Coordinated firing across distant brain regions requires a working metabolism. And the metabolism is precisely what is failing. The blood is gone. The oxygen is gone. The ATP is starting to run out. Yet the brain, in those first seconds, becomes more active and more synchronized rather than less. There are several competing hypotheses about why. One possibility is disinhibition. In the normal awake brain, much of the cortical activity is held in check by inhibitory interneurons, cells that fire to suppress runaway excitation in the network. When energy fails, those inhibitory cells may fail first or fail differently, releasing the rest of the network into a brief, intense burst of uninhibited firing. The brain becomes more active, paradoxically, because the brakes have come off. Another possibility is rebound excitation. As the metabolic substrate fails, ion gradients begin to collapse, and that collapse itself can trigger electrical activity as the system slides through unstable states. A third possibility is something called final network resonance, where the brain, as it loses the ability to maintain its complex dynamic equilibrium, briefly settles into a coherent attractor state that produces synchronized output before everything dissolves. Nobody knows for sure which of these is correct. Probably more than one is operating. What is not in dispute is the data itself. The dying brain, for a window of seconds, does not look like a dying brain. It looks like a brain doing something. And here is the part that connects directly to what we are going to spend this whole video unpacking. Whatever the brain is doing in those seconds, science can measure it from the outside but cannot interpret it from the inside. We can see the gamma waves on a monitor. We can chart their amplitude and their coherence. We can compare them to baseline awake recordings and note that the dying brain produces, by certain metrics, more organized activity in those final moments than in life. But we cannot answer the question of whether anything corresponds to that activity from the perspective of the person whose brain is doing it. The cascade begins not with silence, as the old picture had it, but with a paradoxical surge of organization. And the most active moment in the entire dying process is also the moment from which no report can ever return. Part 2. The human confirmation problem. Most of the early data on this surge came from animal models. The seminal study, often cited in the field, was a 2013 experiment at the University of Michigan that recorded EEG from rats during cardiac arrest and found exactly the kind of gamma surge and cross-frequency coupling I just described. That study was provocative, but it was rats. Skeptics, fairly, asked whether the same thing happens in humans. The answer to that question has been slowly accumulating ever since. The human data is harder to get for obvious reasons. You cannot ethically induce cardiac arrest in healthy people to study what happens. The opportunities to record high-resolution brain activity during natural human dying are rare and require very specific clinical circumstances. Patients who are already in intensive care, already on EEG monitoring, who experience cardiac arrest in the controlled environment of the hospital, or patients undergoing withdrawal of life support, where the dying process is monitored as part of compassionate end-of-life care. Studies in these settings, particularly in the late 2010s and into the 2020s, have begun to confirm that the gamma surge phenomenon is not limited to rats. Bursts of high-frequency activity, resembling the patterns seen in animal models, do appear in human EEG recordings during the dying window. Network reorganization patterns, where different brain regions briefly synchronize in unusual ways, have been documented. The data is still limited. The number of well-documented cases is in the dozens, not the thousands. But the pattern is consistent enough that the phenomenon is now generally accepted as real in human subjects, not just in laboratory animals. Here is the critical limitation, though, and it is the one I want you to hold on to. No surviving patient has ever reported subjective experience corresponding specifically to the dying surge window. Patients who are resuscitated successfully sometimes report what are called near-death experiences, those striking narratives of light, peace, life review, and so on. But the timing of those experiences relative to the EEG events is poorly constrained. The reports come from people who survived, which means by definition the dying process did not complete. We do not know whether the experiences they describe correspond to the gamma surge, to the recovery period, to the moments before cardiac arrest, or to some reconstruction the brain produces afterward. No one has come back from the gamma surge itself, in the strict sense of being recorded during it and then surviving to describe it from the inside. So the scientific community is split. The skeptics interpret the surge as noise, as an artifact of disinhibition with no functional significance for experience. The dying brain is essentially seizing in slow motion, and the gamma activity is the electrical correlate of that seizure, no more meaningful than the random firing patterns you might see in any severely disturbed neural system. The cautious interpreters say the data is unusual enough that we should not dismiss it. Gamma activity in healthy brains is not noise. It is associated with conscious processing. To see the same electrical signature, even briefly, in a dying brain is at least suggestive that some form of organized processing might still be occurring. Whether that processing involves any subjective experience is, of course, the question nobody can currently answer. The ethical constraint is real and it is permanent. We cannot experimentally isolate dying consciousness in humans. There is no experimental design that allows us to test, with controls and replication, what subjective experience is or is not happening during a process that is by definition irreversible and lethal. The closest we can get is observation. An observation in the dying process gives us neural correlates, but not first-person reports. So we are left with this strange tension, and it is the tension that will run through the rest of everything we discuss. The strongest, most organized, most coherent neural activity in the entire dying cascade occurs precisely when the system loses the ability to report what it is doing. The brain becomes most active at the exact moment it can no longer tell us what, if anything, is happening from inside. Part 3. The first structural collapse. The gamma surge is brief. By the second minute after cardiac arrest, the situation in the brain has changed dramatically. The metabolic substrate that supports neural activity is failing in earnest. To understand what comes next, you need to understand a little about how brain cells maintain themselves. Neurons, like all living cells, depend on ion gradients across their membranes. Specifically, they keep sodium concentrations low inside the cell and high outside, and potassium concentrations high inside and low outside. Maintaining these gradients is not free. It costs energy. Specifically, it costs ATP, the molecule that all cellular machinery uses as its energy currency. The pumps that move sodium and potassium across the membrane, the sodium-potassium ATPase pumps, run continuously, consuming ATP to keep the gradients in place. This is one of the most expensive things a neuron does. Estimates suggest that a substantial fraction of all brain energy expenditure goes simply to maintaining these basic ionic gradients. Without those gradients, neurons cannot signal. They cannot generate action potentials. They cannot communicate. They lose, essentially, their identity as functional cells. Now, when the heart stops, blood flow stops. Blood is what carries oxygen and glucose to the brain. Without oxygen and glucose, mitochondria cannot produce ATP. The supply of energy falls catastrophically. Some ATP remains in the cells for a brief window, but it is consumed quickly, and within a minute or two, the supply is essentially exhausted. When ATP fails, the sodium-potassium pumps stop working. The gradients begin to dissipate. Sodium rushes into the cells, potassium rushes out. The membrane potential, which had been carefully maintained at around 70 millivolts negative inside, collapses towards zero. This is what neuroscientists call depolarization. And here is where something genuinely beautiful and terrible happens, depending on how you want to look at it. The depolarization does not happen all at once across the whole brain. It propagates as a wave, specifically as a slow wave called spreading depolarization. Spreading depolarization is a phenomenon that was first described in living, healthy brain tissue back in the 1940s by the Brazilian physiologist Aristides Leão, who discovered it in studies of cortical activity in rabbits. He noticed that under certain conditions, a wave of electrical silence would spread across the cortex at a very specific speed, somewhere between 2 and 5 millimeters per minute. That is unbelievably slow by neural standards. Action potentials propagate at meters per second. Spreading depolarization moves at the pace of a slow continental drift, in neural terms. For decades, spreading depolarization was a curiosity. It was implicated in migraines, where it seems to underlie the visual aura that some migraine sufferers experience. But its full significance only became clear much later, when neurocritical care researchers started to notice it in the brains of patients with traumatic injuries, strokes, and, crucially, dying brains. Spreading depolarization, it turns out, is the wave by which the cortex actually fails when energy is lost. Recent work, particularly by Jens Dreyer and his collaborators, has documented spreading depolarization waves moving across the cortex of dying human patients in the moments after circulation ceases. The wave moves, slowly, across the brain surface. Behind the wave, neurons depolarize fully. Their membrane potentials collapse. Their ability to generate signals is lost. The tissue, in a real functional sense, dies behind the wave as the wave passes over it. Death, when you look at it through this lens, is not a single moment. It is a moving boundary, a front of failure that sweeps across the cortex over a span of minutes, leaving collapsed tissue in its wake. The flat line you see on an EEG monitor, the line that we used to think represented the moment of brain death, is actually the aftermath. It is what you see after the wave has finished moving across the brain. The actual death of cortical tissue is a slow propagating event, not an instantaneous shutdown. This contrasts dramatically with the flatline myth that dominated medical thinking for so long. Death is not a switch, it is a wave. And the wave moves at a pace you could watch with patience if you had the right tools to see it. Now, what does this mean for the central question we are trying to follow? It means that after the gamma surge, after that brief paradoxical burst of organization, the brain transitions into a slow, observable collapse that has its own structure, its own physics, its own characteristic pattern. The cascade does not jump from full activity to silence. It moves through stages, and each stage is visible, measurable, and increasingly well understood, even as the very first stage, the surge itself, remains the single piece of the puzzle that science cannot fully interpret. Part 4. The hidden architecture of collapse. If you zoom into the cellular level during and immediately after the spreading depolarization wave, you see something that looks less like passive failure and more like an active program of self-destruction. The energy crisis triggers a cascade of biochemical events that, taken together, ensure that what just happened is irreversible. The first major piece is glutamate excitotoxicity. Glutamate is the brain's primary excitatory neurotransmitter. Under normal conditions, neurons release controlled amounts of glutamate at synapses, and other neurons respond by firing. The released glutamate is then quickly taken back up by surrounding cells, particularly glial cells called astrocytes, which use ATP to drive the uptake. When ATP fails, the uptake stops. Glutamate accumulates in the extracellular space. The accumulating glutamate continues to bind to receptors on neurons, particularly NMDA receptors, which are calcium-permeable channels. The constant stimulation forces calcium to flood into the neurons in massive quantities. Calcium, in small, controlled amounts, is a critical signaling molecule. In large, uncontrolled amounts, it is a poison. The calcium overload triggers a cascade of destructive enzymes. Calpains and other proteases begin to chew through the cellular cytoskeleton. Lipases attack the cell membrane. Endonucleases begin to fragment the DNA. The mitochondria, already failing from the loss of oxygen, undergo a process called mitochondrial permeability transition, where their membranes lose integrity and they release a flood of pro-death signaling molecules into the cell. Some of these molecules trigger apoptosis, like cascades, programmed cell death pathways that, in a healthy organism, are used to remove damaged cells in a controlled way. In the acute setting of cardiac arrest, full apoptosis usually does not have time to complete in its classical form, but the partial activation of these pathways contributes to the broader destruction. The cytoskeleton breaks down. Synapses, those exquisitely complex structures that store the actual information of memory and personality and learned behavior, begin to disintegrate. The intricate molecular machinery that took the brain decades to build is dismantled in minutes. This is critical to understand. The brain is not just losing power, it is being actively disassembled by its own internal machinery that was held in check throughout life by the constant energetic effort to suppress it. When the energy fails, the suppression fails, and the destructive programs run to completion. You can think of it, with some poetic license, as a computational system crash, where each cascading subsystem failure triggers further failures, a kind of digital decoherence, where every level of the system pulls down the levels below it. The information encoded in the brain, in its synaptic connections, and its electrical patterns, and its molecular states, has no time to be preserved. Within minutes, the substrate that held it is gone. And here is what is worth pausing on for a moment. The active nature of this collapse is part of why brain death is so different from other forms of organ failure. The brain does not just stop, it dismantles itself. The structures that held identity, memory, personality, perception, all of those functions, are not simply paused. They are torn down at the molecular level by enzymes that were always there, always ready, always held in check by metabolism that no longer exists. Once the cascade has run, there is no putting it back. There is no thawing the brain like a frozen system and recovering its previous state. The information is gone because the machinery that encoded it has been chemically taken apart. So the cascade we are following, from the gamma surge through spreading depolarization through this biochemical demolition, is not a passive fading. It is an active, irreversible program. The brain dies the way a building demolishes when the load-bearing structures are cut all at once. There is an order to the collapse. There is a sequence. And the sequence has the strange feature of being most organized and most coherent at its very beginning, in that gamma surge window, before the cascade of self-destruction really begins to bite. By the time the spreading depolarization wave has finished its slow sweep, the brain that produced the surge is gone. The structures that might have supported any subjective experience corresponding to that surge are gone. Whatever happened in those first 30 seconds, if anything happened from the inside, has no surviving substrate from which to be reported. And the body, meanwhile, is just beginning its own much longer transformation. Part 5. The first hour after identity ends. Once the brain has finished its collapse, the conventional definition of clinical death is satisfied. The heart is not beating, the brain is not functioning, the person, in any practical or medical sense, is gone. But here is the thing that most people never think about, and that I find genuinely one of the stranger facts in all of biology. The body is not gone, not even close. The body continues to be a complex, dynamic, chemical and physical object for hours, days, weeks, and in some senses, years afterward. The cascade does not stop with the brain. It just shifts to a different layer of the system. And if you want to understand what death actually is, as a physical process rather than a moment, you have to follow it all the way down. The first thing that happens after circulation stops is almost invisible from the outside. The cells of the body, suddenly deprived of the oxygen that blood was delivering, begin to switch metabolic strategies. Most of your cells, under normal conditions, run on aerobic metabolism. They burn glucose in the presence of oxygen and produce ATP very efficiently, the way a well-tuned engine burns fuel cleanly. But many tissues have a backup system, They can shift to anaerobic metabolism, a more primitive and far less efficient process called glycolysis, which squeezes a small amount of ATP out of glucose without needing oxygen at all. It is not elegant. It is the metabolic equivalent of burning wet wood when the gas runs out. It works, briefly, but it produces a problematic byproduct, lactic acid. The lactic acid begins to accumulate in the tissues, lowering the local pH, making the cellular environment more acidic, and contributing to the eventual breakdown of everything that depends on carefully maintained chemical balance, which, in the body, is essentially everything. Now, here is something worth pausing on, because it genuinely surprised me the first time I looked into it. Different tissues fail at vastly different rates. We tend to think of death as a single event, a switch that flips, a light that goes out. But that is not what the biology looks like at all. The brain, as we discussed earlier, collapses within minutes of losing blood flow. Cardiac muscle, the tissue that makes up the heart itself, fails relatively quickly as well, within tens of minutes to a few hours, depending on temperature and conditions. Skeletal muscle, the tissue in your arms and legs and torso, is more durable than either of those. Skin and connective tissue can remain biologically viable for many hours after the organism is officially dead. And some cells, certain skin cells, various stem cell populations tucked into tissues throughout the body, can survive for more than a day after clinical death. There are documented cases of skin cells being successfully cultured in the laboratory from cadavers more than 24 hours after the person died. 24 hours. The cells are still alive. They can still divide. They still have metabolic activity. The person is gone, but the colony they were built from is still running quietly on its own internal reserves, completely unaware that the central authority has collapsed. This is actually how we should be thinking about the body in the first place. You are not a single organism in a simple sense. You are a colony, a vast cooperating community of around 37 trillion human cells, each one a semi-autonomous living unit with its own metabolism and its own survival timeline. What we call death is really the dissolution of the colonial arrangement, the collapse of the coordination system that held all those cells in a unified, purposeful relationship with each other. The cells themselves do not all get the memo at the same time. Some of them keep going, keep burning their reserves, keep doing what they have always done, long after the organism that coordinated them has ceased to exist. Death, at the cellular level, is not a moment. It is a slow, staggered process that plays out across a timeline of hours to days. Externally, the changes are more obvious, and they happen in a sequence that forensic pathologists have studied carefully enough to use as a clock. The first and most immediate change is called pallor mortis, the paleness that appears within minutes of death as blood circulation stops and the small surface capillaries lose their pressure. The warm pink of living skin, maintained by constant blood flow, drains away. The skin becomes pale and waxy. This is one of the first things people notice when they see a recently deceased body, and it happens fast, within minutes of the heart stopping. Then comes algor mortis, the cooling of the body. A living body is a heat engine. It produces metabolic heat as a constant byproduct of cellular activity, and it maintains its core temperature at around 37 degrees Celsius by balancing heat production against heat loss to the environment. When metabolism stops, heat production stops, and the body begins to cool toward the ambient temperature of the room it is in. The rate of cooling is roughly 1.5 degrees Celsius per hour under typical indoor conditions, though the actual rate varies with body size, clothing, and the temperature of the surroundings. It slows down as the temperature difference between the body and the room narrows, following a basic thermodynamic curve. Forensic pathologists use this curve, they call it the post-mortem cooling curve, to estimate how long ago death occurred, though it is one of several indicators rather than a precise instrument on its own. And then there is livor mortis. This one is stranger looking than the others, and it takes a few hours to develop fully. Without a heart to pump blood around, gravity takes over. Blood is a liquid, and like any liquid, it flows downward. In a body lying on its back, blood begins to pool in the lowest accessible regions, the back, the buttocks, the backs of the legs. The pooled blood produces a reddish-purple discoloration of the skin in those areas, while the areas under pressure, where blood is physically squeezed out of the capillaries by the weight of the body, remain pale. The pattern of livor mortis is fixed in place after a certain number of hours, as the blood clots and the discoloration becomes permanent. If a body has been moved after death, the livor pattern tells the story if the discoloration is on the back, but the body is found face down. Something changed. Forensic investigators read livor mortis the way you might read a map. None of this is the brain doing anything. All of it is happening because the brain is no longer running the show. The body has been handed over completely to physics and chemistry. It is now a passive object, a warm, complex, slightly acidic object sitting in a room, subject to the same gravitational and thermodynamic laws that govern any other mass in a gravitational field. Gravity pulls the blood down. Thermodynamics pulls the heat out. Chemistry is already beginning to do its slower, quieter work on the inside. And that internal chemistry is worth dwelling on for a moment, because from the outside, the body looks completely static. The skin pales, the limbs cool, the chest does not rise. To anyone watching, it looks like nothing is happening. The absence of process is total and obvious, but that appearance is an illusion, and it is one of the things I find genuinely fascinating about the post-mortem period. Internally, on a molecular and cellular level, the body is changing constantly, and the rate of change in the first several hours is significant. Cells that still have metabolic capacity continue burning through their internal energy reserves. Mitochondria that still have substrate available keep producing ATP for as long as they can. Enzymes that spent the entire life of the organism carefully contained within membrane-bound compartments, lysosomes, the little cellular organelles whose whole job is to digest things, begin to leak as those membranes degrade without energy to maintain them. When lysosomal enzymes escape into the cell interior, they begin to digest the cell from within. This process has a name, autolysis, or self-digestion. It is entirely chemical, requires no bacteria, no external agents, just the body's own molecular machinery running loose in an environment where there is no longer any control. The pH of various tissues shifts as lactic acid accumulates, and enzymatic reactions alter the local chemistry. The carefully maintained electrochemical gradients that defined every cell's interior are slowly dissipating. The molecular architecture of the body is quietly beginning to come apart, one compartment at a time, largely invisible from the outside. There is something philosophically interesting buried in this. The process that begins in those first hours, autolysis, pH drift, the leaking of contained enzymes, is not death arriving from outside. It is the body's own machinery, always present, always capable of this, running for the first time without supervision. The destructive potential was always there. Life, in a sense, was the ongoing project of keeping it in check. When the energy runs out and the control systems fail, the latent chemistry of dissolution simply begins to express itself. The body was always capable of doing this. It just never had the chance before. So the cascade that began with the gamma surge in the first 30 seconds has not ended. It has shifted venues, changed timescales, moved from electrical to chemical, from milliseconds to hours. The same structured sequential transformation is still running. It has just slowed down enough that you have to look more carefully to see it. And what comes next is where it gets considerably more dramatic. Part 6. The locking and unwinding of muscle. A few hours after death, sometimes as soon as 2 or 3, sometimes as long as 6 or 7, depending on the temperature of the environment, and the physical condition of the person, the body begins to stiffen, not dramatically, not all at once. It starts quietly in the smallest muscles and then spreads over the course of hours through the entire muscular system until the body has become something closer to a rigid object than a flexible one. This is rigor mortis, and it is probably the most universally recognized sign of recent death in all of human history. Every culture that has ever dealt with bodies has encountered it. Every tradition of handling the dead has had to work around it or account for it. And for most of that history, nobody really knew what was causing it. The folk explanations were understandable given the information available. People assumed the stiffening was some kind of final muscular contraction, the body seizing up in its last protest, or tension accumulating in tissues that no longer had the nervous system's permission to relax. Some early medical writers thought it might be related to the coagulation of the blood, or to some kind of chemical hardening of the muscle fibers themselves, like meat setting in the cold. The stiffness felt so purposeful, so structured, that it seemed like it had to be an active process, something the body was doing rather than something the body was failing to do. The actual explanation, when it was finally worked out at the biochemical level, turned out to be almost exactly the opposite of what people had assumed. Rigor mortis is not the body contracting, it is the body failing to relax. And the difference between those two things is the entire story. To understand it, you need to know a little about how muscles actually work. Muscle tissue is built around two proteins, actin and myosin. Actin forms long, thin filaments that run through the muscle fiber. Myosin is a larger, heavier protein with a head structure that can reach out and physically grab onto the actin filament. When a muscle contracts, myosin heads attach to actin, pull, detach, and attach again further along a kind of molecular ratchet, cycling through attachment and release thousands of times per second, with each cycle driving the filaments past each other and shortening the muscle as a whole. It is an extraordinarily elegant piece of molecular machinery, refined over hundreds of millions of years of evolution. Here is the critical detail. Every step of that cycle, the attachment, the pull, the release, requires energy. Specifically, it requires ATP. And the step that matters most for understanding rigor mortis is the release step. Myosin can attach to actin spontaneously without ATP. It does not need energy to grab on. What it needs energy for is to let go. ATP binds to the myosin head and causes a conformational change that releases it from the actin filament, allowing the cycle to reset and the muscle to relax. Without ATP, myosin grabs actin and stays there. The molecular handshake becomes permanent. The crossbridge is locked. So what happens in the hours after death is straightforward once you see it. ATP production stops when metabolism stops, but there is still some ATP in the muscle cells at the moment of death, a small reserve. For the first hour or two after death, that reserve is enough to keep the actin-myosin crossbridges cycling normally, which is why the body remains relatively soft and flexible in the very early post-mortem period. The muscles are not producing force because there is no nervous signal telling them to contract, but they can still release and reset. They can still relax in the mechanical sense. As the hours pass, the ATP reserve depletes. More and more myosin heads, as they cycle through attachment, find themselves without the ATP needed to detach. They lock onto actin and stay there. The muscle stiffens progressively, fiber by fiber, sarcomere by sarcomere, until the entire muscle group has become rigid. The process moves through the body in a characteristic order, starting with the small muscles of the eyelids and jaw, which have a higher ratio of surface area to volume and deplete their ATP faster, and then spreading progressively to the larger muscle groups of the neck, the arms, the trunk, and finally the legs. Peak stiffness is typically reached somewhere around 12 hours after death. At that point, the body is genuinely rigid. Moving a limb requires force. The joints resist. Everything has locked into whatever position it happened to be in when the ATP ran out. And then, after another 12 to 24 hours, the rigor begins to release. The body softens again. The joints become movable. At first glance, this sounds like the process reversing, like the muscle is somehow recovering. It is not. What is happening is far more final than that. The proteins themselves are beginning to break down. The same autolytic enzymes we discussed earlier, freed from their cellular compartments as membranes degrade, have been working on the muscle proteins throughout this time. By the time 36 to 48 hours have passed, enough of the actin and myosin have been fragmented by enzymatic digestion that the cross bridges are physically falling apart. The lock releases not because the key has been found, but because the lock itself is dissolving. The body softens into the beginning of structural decomposition at the muscular level. What feels like relaxation is actually the tissue beginning to liquefy. This sequence, stiff, then soft, again has caused genuine confusion throughout history, and not just among non-specialists. Before the biochemistry was understood, the two-phase pattern of rigor was a puzzle. The same body that was rigid yesterday is pliable today. What had changed? Some historical accounts of bodies apparently moving or being found in different positions than expected may have roots in this phenomenon, combined with a poor understanding of how gases building up in decomposing tissue can shift a body's posture over time. The gas-driven movements are real. A body lying in a certain position, as decomposition gases accumulate in the abdominal cavity and elsewhere, can shift, settle, or even briefly produce sounds as gas escapes through natural openings. None of this is subtle if you are in an enclosed space with a decomposing body, and none of it has anything to do with life. It is chemistry and physics doing exactly what chemistry and physics do with any organic material that is breaking down and releasing gaseous products. There is also the matter of post-mortem nerve responses, which are genuinely strange and have contributed to historical anxieties about premature burial and apparent revival. Peripheral nerves, the nerves in your limbs and torso, as opposed to the central nervous system in the brain and spinal cord, retain the ability to propagate electrical signals for some time after death. The exact window varies, but in the first few hours, peripheral nerves can still respond to external stimulation. If you apply an electrical stimulus, or even a strong mechanical one to a nerve in the arm of a recently deceased person, the muscle it innervates may twitch. The response is a reflex arc that does not require the brain. It is a local electrical event in a wire that happens to still be functional, connected to a motor that happens to still have enough intact machinery to respond. It means nothing. It is not life. It is not pain. It is not awareness. It is a residual electrical property of tissue that has not yet fully degraded. But it looks startling, and in historical contexts without biochemical knowledge, it was terrifying. So rigor mortis, in the full context of the cascade we have been following, is the muscular system's version of exactly what happened in the brain. The brain ran out of ATP, the ion pumps failed, the membrane potentials collapsed, and spreading depolarization swept across the cortex in a slow wave of electrochemical failure. The muscles run out of ATP, the myosin heads lock onto actin, and a slow wave of mechanical rigidity spreads from the small muscles to the large ones. Different tissue, different mechanism, different timescale, but the same underlying logic. The same fundamental sentence being written in a different molecular language. When the energy stops, the dynamic processes that depended on energy lock into their lowest available state. Structures that were held in flexible, active configurations by continuous metabolic input settle into rigid, stable, low-energy configurations instead. The thermodynamics do not care whether they are operating on neurons or muscle fibers. The principle is identical. The energy was the only thing keeping everything moving. Remove the energy, and everything stops not in the same way or on the same schedule, but inevitably in the same direction. Layer by layer, the body is expressing the same truth, and it is almost elegant, in a cold sort of way, to see how consistent that truth is across scales. From the millisecond firing of a neuron to the hours-long stiffening of an arm, the cascade has a single underlying theme. Metabolism is what separates the living state from the unliving one, and without it, the complex, improbable architecture of a human body begins its long, structured, patient return to equilibrium. The muscles have now had their say. What comes next is louder, stranger, and involves considerably more participants. Because the body is about to stop being an object and start being an address. Part 7. The Microbial Takeover. Here is something worth sitting with for a moment before we go any further. Right now, as you watch this, somewhere between 30 and 40 trillion bacterial cells are living inside your body. That number is so large it barely registers. So let me put it another way. There are roughly as many bacterial cells in and on you as there are human cells making up your actual tissues and organs. You are, by cell count, approximately half microbial. You have been your entire life. And for your entire life, you have been almost completely unaware of it, because the arrangement has been working smoothly enough that it never demanded your attention. Most of those bacteria live in your digestive tract, particularly in the large intestine, where conditions of warmth, moisture, and regular nutrient delivery have made your gut one of the most densely populated microbial environments on the planet. Others colonize your skin, your mouth, your nasal passages, the lining of your respiratory tract, essentially every surface of your body that has any contact with the outside world carries its own characteristic microbial community, shaped by the local chemistry and the local conditions. The communities in different parts of your body are as distinct from each other as different forests on different continents. Your mouth bacteria and your gut bacteria and your skin bacteria are not the same populations. They are different ecosystems occupying different niches within the larger ecosystem of you. Throughout your life, this arrangement has been held in place by 3 overlapping systems. The first is your immune system, an extraordinary surveillance and response network of white blood cells, antibodies, chemical signals, and physical patrol mechanisms that monitors every surface and cavity of your body and contains any microbial population that tries to expand beyond its permitted territory. The second is the chemical environment of your tissues, the pH, the oxygen levels, the specific molecular composition of each region, which creates conditions that favor certain bacteria in certain places and suppress others. The third is physical barriers, the tight junctions of the gut lining, the layers of your skin, the mucous membranes of your respiratory tract, physical structures that physically separate microbial populations from the tissues they would otherwise colonize freely. The relationship is mostly symbiotic, and we should be clear about that before going any further. This is not a story of parasites being politely tolerated. The bacteria in your gut help you digest certain foods your own enzymes cannot handle, produce vitamin K and some B vitamins that your body uses, help calibrate your immune system during development, and contribute to a range of metabolic processes that we are still in the early stages of fully mapping. In return, they get an extraordinarily stable, warm, well-supplied environment. It is a genuine partnership, refined over millions of years of co-evolution. You need them. They need you. The arrangement works. When you die, the arrangement ends. And it ends faster than almost anything else in the cascade. Within minutes of the cessation of circulation, the immune system stops functioning. White blood cells are metabolically expensive to maintain and to move. They run on ATP like everything else, and without blood delivering oxygen and glucose, they run out of fuel very quickly. The cells that normally patrol your tissues, identify intruders, and coordinate responses to any breach simply stop. The chemical signals that maintained territorial boundaries between microbial communities dissipate as the cells producing them die. The tight junctions of the gut lining, which are active structures requiring energy to maintain their integrity, begin to fail. As the cells that built them lose the ability to hold themselves together, the physical barriers come down, the chemical environment shifts as pH drops, oxygen disappears, and the carefully maintained gradients of the living body dissolve into something much more uniform and much more permissive. The bacteria, particularly the anaerobic bacteria that dominate the lower gut, find themselves in a situation that has no precedent in their experience. The walls are coming down, the guards are gone, and stretching out in every direction is an enormous quantity of warm, nutrient-rich tissue that nothing is currently defending. They move, not with any intention or awareness. Bacteria do not have those things, but in the way that any population expands when resources become available and constraints are removed. The gut bacteria begin to cross the intestinal barrier and spread into the surrounding tissues. They move through the mesentery, the connective tissue that holds the intestines in place. They spread into the abdominal cavity. They begin to colonize the organs they encounter, consuming them as they go. This process is called putrefaction, and it is, alongside rigor mortis, one of the most studied and most visible phases of decomposition. It is also, by most human metrics, the most confronting. The chemistry of putrefaction produces a characteristic set of compounds, some of which have become almost culturally famous. Cadaverine and putrescine are probably the most well-known, and their names are not accidental. These are compounds whose smell has been shaping human responses to death for as long as humans have existed. They are both diamines, produced when bacteria break down certain amino acids from proteins in the decomposing tissue. Specifically, cadaverine comes from the amino acid lysine and putrescine from ornithine, which is itself derived from arginine. These molecules are not unique to decomposition. Small amounts are produced in normal metabolic processes in living organisms. But in decomposition, they accumulate in quantities that produce the distinctive and overwhelming odor that every predator, scavenger, and carrion insect in the history of life has been evolutionarily tuned to detect. Sulfur compounds are another major product. Hydrogen sulfide, the compound responsible for the smell of rotten eggs, is produced in significant quantities as bacteria break down sulfur-containing amino acids like cysteine and methionine. It contributes both to the odor and to visible changes in the appearance of the tissues. The greenish-black discoloration that develops during active putrefaction is partly due to sulfur compounds reacting with iron from SPEET, hemoglobin in the blood to form iron sulfide, which is dark-colored and accumulates in the skin and underlying tissues. And then there is the gas. As bacteria metabolize the tissues, they produce gases as byproducts: carbon dioxide, methane, hydrogen sulfide, ammonia, and others depending on the specific bacterial species and the substrate they are consuming. These gases have nowhere to go except to accumulate. They fill the body cavities. The abdomen begins to distend. In warm conditions, where bacterial activity is fastest, the bloating can become extreme. Within a few days, the abdomen swells dramatically. Fluids are forced out of the body's natural openings. The face becomes distorted and unrecognizable. The internal pressure builds until it is sufficient to rupture tissues from the inside. This is the bloat stage of decomposition, and it is the most visually dramatic transformation the body undergoes after death. It is also, in a way, the high water mark of the microbial takeover, the moment when the scale of the bacterial activity becomes visible from the outside in unmistakable physical form. While this internal transformation is accelerating, something is also happening at the surface. The chemical signatures of early decomposition, the volatile organic compounds, the gases, the sulfur compounds diffusing through skin broadcast a signal into the surrounding environment. And that signal has receivers that have been tuned to it by millions of years of evolution. Blowflies are typically among the very first to respond. Different species of blowfly are sensitive to slightly different chemical signatures and arrive at different stages. But the earliest arrivals can detect the odors of decomposition from impressive distances and can locate a body within minutes to hours of exposure to the air, depending on conditions. They lay their eggs in batches, preferentially in moist areas, the eyes, the mouth, the nostrils, any wound or opening, and the eggs hatch into larvae within hours to a day or two. The maggots feed on soft tissue with remarkable efficiency, and a large enough mass of maggots can strip a body of its soft tissue in a matter of days under the right conditions. After the blowflies come other fly species, then beetles, then a succession of arthropods that each prefer a different stage of decomposition, some that favour fresh tissue, others that prefer partially dried material, others that specialise in the drier, more skeletonized remains of later stages. The sequence is predictable enough that forensic entomology has become a genuine discipline capable of estimating time of death from the species present and their developmental stages with an accuracy that can, in favourable cases, reach within hours over spans of days or weeks. And here is the conceptual shift I want to land carefully, because I think it is one of the more genuinely interesting framings in this entire cascade. The body, at this point in the process, has stopped being an organism. It has become an ecosystem. The boundary between you and not you, the boundary that your immune system, your skin, your gut lining, and your mucous membranes spent your entire life defending and maintaining has dissolved. There is no longer a you that those systems are protecting. There is just a substrate, a physical and chemical environment with particular properties of temperature and moisture and nutrient availability, in which a complex, multi-species, ecologically structured succession of organisms is playing out according to rules that have nothing to do with the person who used to be there. The bacteria that were always inside you are now consuming you. The flies that always existed in the environment around you are now using your tissues as a nursery for their offspring and a food source for their larvae. You have gone from being a host to being a habitat. This is not a poetic metaphor. It is a literal ecological description of what is happening. The cascade that began with 30 seconds of organized gamma wave activity in a dying brain has now expanded outward across days and weeks to include a succession of species, each one responding to chemical signals and physical conditions, each one giving way to the next as the substrate changes, each one following rules laid down by evolution over timescales that dwarf the entire history of our species. The architecture of the cascade is still there. The structure is still there. It is just scaled up from neurons to bacteria to flies to beetles, from milliseconds to hours to days to weeks, and it is still moving, still predictable, still obeying the same fundamental logic it has obeyed since the very first second. The system is going somewhere, and it is not done yet. Part 8. The body becomes a weather system of chemistry. By the time decomposition has been running for a few weeks, something has shifted in the nature of what is happening. The bacterial takeover we talked about in the last section is still ongoing, still consuming soft tissue, still producing its characteristic chemistry. But a second process has been running alongside it the entire time, quieter and less visible, and by this stage it has become just as significant. It does not involve bacteria at all. It does not involve any living organism. It is purely chemical, and it has been building since the very first minutes after death, waiting for the structural barriers of the living cell to fail enough to let it run freely. The process is called autolysis. The word comes from the Greek for self-digestion, and that is exactly what it is. Your body, throughout your life, has been carrying the enzymatic machinery capable of dissolving itself. Locked away inside membrane-bound compartments within every cell, particularly in organelles, called lysosomes, are powerful digestive enzymes, proteases that break down proteins, lipases that break down fats, nucleases that fragment DNA and RNA, carbohydrases that attack carbohydrates. These enzymes spend your life performing essential maintenance functions, breaking down damaged proteins, recycling cellular components, digesting waste materials, managing the constant turnover of molecular machinery that keeps cells functional. They are indispensable. They are also, if they ever escape their compartments, catastrophically destructive. Living cells keep these enzymes carefully contained. Lysosomal membranes are actively maintained structures, requiring energy to hold together and to regulate what passes through them. As long as the cell is alive and metabolically active, the membranes hold and the enzymes stay where they belong. When the cell dies and ATP production stops, the membranes begin to lose integrity. They become permeable. Then they rupture. The enzymes spill out into the interior of the cell, and then, as cell membranes fail in turn, into the surrounding tissue. And once they are loose, they do what they were always capable of doing. They digest whatever they encounter. This is worth pausing on, because I think it reframes the entire story of decomposition in an interesting way. We tend to think of a body decomposing as something being acted upon from outside, by bacteria, by insects, by the environment, and those external agents are real and important. But autolysis reminds us that the body is also being dissolved from within by its own chemistry, using machinery that was always present and always capable of this. The destructive potential was built into every cell from the beginning. Life was the ongoing project of keeping it contained. Death is what happens when the containment fails, and the self-digestion that was always latent in the system finally runs without supervision. Autolysis begins almost immediately after death. The very first enzyme leakage from the earliest dying cells starts within the first hour, but it takes days to weeks to become macroscopically significant, because the initial leakage is limited and the process builds gradually as more and more cells fail and release their enzymatic contents. By the second and third week, in typical conditions, autolysis has become a major driver of tissue breakdown running in parallel with bacterial putrefaction. The two processes reinforce each other. Autolysis breaks down cellular structures, making tissues more accessible to bacterial colonization. Bacterial metabolic products alter the pH and chemical environment of the tissues, which in turn affects the activity of autolytic enzymes. The two processes are not separate stories. They are a single coordinated dissolution, chemical and biological, working together toward the same endpoint. The result, over weeks to months, is liquefaction. Soft tissues, the organs, the muscles, the connective tissues, the fat, progressively lose their structural integrity and merge into an increasingly homogeneous chemical mixture. The liver, which is particularly rich in digestive enzymes, often autolyses faster than other organs and may be largely liquefied while the skin is still partially intact. The brain, also enzyme-rich and highly lipid-heavy, breaks down quickly. Muscle tissue, more structurally robust, takes longer but eventually follows. The distinct anatomical structures that define the body's interior, the boundaries between organs, the layering of tissues, the precise architecture of vessels and ducts dissolve. What remains is not recognizable as the interior of a human body. It is a complex chemical mixture, variable in its composition from one region to another depending on what was there before, but no longer organized into the structures that gave it meaning. The fluids that this process produces do not stay in place. They redistribute through whatever pathways are available through tissue spaces, along fascial planes, toward the body's exterior and into the surrounding environment. A body lying on soil becomes the source of a slow seep of chemically complex fluid into the ground beneath it, a phenomenon that soil scientists and forensic ecologists have studied extensively. This fluid, sometimes called cadaver decomposition fluid, carries with it an extraordinary load of dissolved organic compounds, nitrogen, phosphorus, sulfur, and a range of other chemicals derived from the breakdown of tissues. It alters the soil chemistry in ways that can persist for years after the body itself is gone. The microbial community in the soil beneath a decomposing body changes dramatically, responding to the sudden influx of nutrients. Plant growth in the immediate area is often affected, sometimes enhanced by the nutrient loading, sometimes suppressed by the chemical toxicity of specific decomposition compounds, sometimes both in sequence as the chemistry changes over time. Botanists and forensic ecologists can sometimes identify the location of a former grave years or even decades later simply by looking at the vegetation pattern and the soil chemistry. Gases continue to be produced throughout this period: carbon dioxide, methane, hydrogen sulfide, ammonia, and others. And as the soft tissues liquefy, the structural containment that had been trapping them breaks down. The bloated phase of decomposition, driven by gas accumulation in intact body cavities, gives way to a deflation as those cavities rupture or find pathways for the gas to escape. The body flattens. The dramatic distension resolves into a spreading pool of liquid and semi-solid material. The outer structures, skin, hair, nails, which are made of more chemically resistant proteins than the soft tissues, persist longer, but eventually follow. Keratin, the protein that makes up hair and nails and the outer layers of skin, is notably resistant to many proteases, which is why these structures often remain recognizable long after the soft tissues beneath them have dissolved. But by this stage of decomposition, they have lost their underlying support. Hair detaches in clumps, nails separate from the digits. Skin, no longer adhered to the tissues it once covered, slides and detaches. The face, which in life was the primary site of identity, recognition, expression, and connection, becomes, in the most literal possible sense, unrecognizable. Not damaged or distorted in the way a wound distorts a face, but chemically transformed into a surface that no longer carries any of the structural information that made it a face in the first place. It is a chemical surface now, a former face. The identity it held has no more physical substrate left to cling to. From a physics standpoint, all of this is the second law of thermodynamics, expressing itself at the scale of a human body. And I think it is worth spending a moment with that framing, because it connects what we are watching to something genuinely fundamental about the universe. The second law says, broadly, that the entropy of a closed system tends to increase over time. Entropy, roughly speaking, is a measure of disorder, or more precisely, a measure of how many different ways the components of a system could be arranged. A highly ordered system, with its components in specific precise configurations, has low entropy. A disordered system, with its components distributed in less specific, more probable arrangements, has high entropy. The universe tends toward higher entropy over time because high entropy states are simply more probable than low entropy ones. There are vastly more ways for things to be disordered than there are for things to be ordered. So in the absence of forces maintaining order, disorder is where systems end up. A living human body is one of the most extraordinarily low entropy objects in the known universe. The molecules that make it up are arranged in configurations of almost unimaginable specificity: folded proteins in precise three-dimensional shapes, DNA sequences encoding information in exact nucleotide order, membranes with specific lipid compositions, organelles positioned in precise relationships to each other, the whole architecture maintained against the thermodynamic current by continuous energy input from food. Maintaining this state of low entropy is expensive. It is, in a fundamental sense, what eating is for. Food is the energy input that allows the body to keep holding itself in its improbably ordered state against the constant thermodynamic pressure toward disorder. When that energy input stops, thermodynamics wins, immediately, inevitably, completely. The ordered structures break down into less ordered components. The less ordered components mix with each other and with the environment. The carefully separated chemical compartments merge into a single medium. the specific molecular configurations dissolve into more probable, more generic chemistry. The body, which during life was a tightly bounded, highly specific, energetically maintained chemical system, becomes an open system, exchanging material freely with the soil and air and water around it, losing its boundaries, losing its specificity, becoming, gradually and then completely, indistinguishable from the chemistry of the environment it is decomposing into. What you are left with, by the end of this stage, is not a body. It is not even the remnants of a body in any meaningful structural sense. It is a region of altered chemistry, a patch of soil and air that is somewhat different from the surrounding soil and air because of what was once there, a chemical ghost, a weather system of organic compounds dispersing outward into the larger systems of the planet, carrying with them the atoms that once, in a very different arrangement, constituted a person. Those atoms are going somewhere. They have been going somewhere this entire time. And the distances they are about to travel, and the timescales on which they will travel them, are about to expand in ways that make everything we have discussed so far look like a very small and very local event. Part 9. Skeletons, DNA, and time that outlives biology. After the soft tissues have gone, after the weeks and months of bacterial putrefaction and autolytic liquefaction have done their work, what remains is the skeleton. And the skeleton is a completely different kind of object from everything we have been discussing. Everything that came before in this cascade happened fast, by geological standards. The neural collapse in minutes, the cellular shutdown in hours, the soft tissue decomposition in weeks to months. The skeleton operates on a different clock entirely. It is built from different materials, governed by different chemistry, and it persists on timescales that begin to blur the boundary between biology and geology. To understand why, you need to know what bone actually is, because most people have a vague mental model of bone as something like dry white rock, inert and simple. The reality is considerably more interesting. Bone is a composite material, and one of the most sophisticated composite materials that biology has ever produced. It is built from two primary components that, on their own, would each be inadequate for the job bone has to do, but together produce something with properties that neither possesses alone. The first component is hydroxyapatite, a calcium phosphate mineral with the chemical formula Ca10PO4CnOH2. Hydroxyapatite is what gives bone its hardness and its compressive strength, its ability to bear weight without crushing. In its pure form, however, a material made entirely of hydroxyapatite would be brittle. It would bear weight well, but shatter under impact, the way ceramic does. That is where the second component comes in. Collagen is a fibrous structural protein, the same protein that gives tendons and ligaments and skin their tensile strength. In bone, collagen fibers run through the hydroxyapatite mineral matrix in a precise, organized arrangement, giving the composite material its flexibility and its resistance to fracture. The mineral resists compression. The collagen resists tension and absorbs impact. Together, they produce a material that is both hard enough to support the body's weight and tough enough to withstand the physical stresses of a life in motion. It is, by engineering standards, a remarkably elegant solution to a genuinely difficult materials problem. Engineers designing synthetic bone substitutes have spent decades trying to replicate it and have not fully succeeded. This composite structure is also what determines how bone behaves after death and why its two components have such different persistence timescales. Collagen, being a protein, is vulnerable to the same biological and chemical processes that break down soft tissues, just more slowly. Bacteria can digest collagen given enough time. Water hydrolyzes its peptide bonds. Heat accelerates the breakdown. In warm, wet, oxygen-rich environments, the conditions that are most favorable for rapid soft tissue decomposition, collagen, begins to degrade meaningfully within years to decades after death, and may be substantially gone within a century. As the collagen matrix breaks down, the bone loses its flexibility and toughness. It becomes more brittle, more prone to cracking and fragmenting under physical stress. Old bones from warm, wet burial environments often crumble when handled, precisely because their collagen is gone and only the mineral matrix remains. And the mineral matrix without its collagen reinforcement is fragile. The hydroxyapatite mineral component is a different story. Minerals are not organic molecules. They do not have peptide bonds to hydrolyze or carbon chains to oxidize. They are stable crystal lattices of calcium, phosphate, and hydroxide ions, and they can persist in that form for extraordinarily long periods. In dry, cool, alkaline environments, conditions that minimize both biological activity and chemical weathering, hydroxyapatite from bone can remain structurally intact and chemically recognizable for thousands of years. Bones recovered from desert environments, from cave deposits, from permafrost, can be thousands of years old and still retain their original structure in remarkable detail. The mineral is genuinely durable in a way that biological materials almost never are. And then there is fossilization, which is a different phenomenon entirely and one worth spending some time on, because it represents the extreme end of the skeleton's persistence timeline and connects the story of one body to the deep history of life on Earth. Fossilization is not simply preservation, it is replacement, under very specific conditions, typically involving rapid burial in sediment that is rich in dissolved minerals, combined with the gradual percolation of mineral-laden groundwater through the buried remains over geological time. The original material of the bone is slowly replaced, molecule by molecule, with minerals from the surrounding environment. The calcium phosphate of the hydroxyapatite may be replaced with silica, or with calcite, or with iron compounds, or with any of a range of other minerals, depending on the local geochemistry. The result is a rock that preserves the shape and often the microscopic structure of the original bone in extraordinary detail, but is made of entirely different material. The bone is gone. The fossil is a mineral replica of the bone, preserved in stone. Fossilization requires an improbable combination of circumstances. The organism has to die in or near a depositional environment where sediment will cover the remains quickly before scavengers and weathering can destroy them. The sediment chemistry has to be right for mineralization. The burial has to be deep enough and the conditions stable enough for the mineralization to proceed over millions of years without the fossil being destroyed by geological processes, uplift, erosion, metamorphism. The vast majority of organisms that have ever lived left no fossil record whatsoever. Their remains decomposed completely and rejoined the chemical cycles of the planet without a trace. The fossil record is not a sample of life. It is an extreme rarity, the tiny fraction of organisms whose deaths happen to coincide with the right place, the right sediment, and the right chain of geological luck to produce a mineralized replica that survived to be found. But when it works across timescales that are almost impossible to hold in your head. The oldest, reliably dated animal fossils are around 600 million years old. There are microbial fossils approaching 3.5 billion years old. The mineral replica of a body can, under the right conditions, outlast the continent it was buried in, surviving multiple cycles of mountain building and erosion, of ocean opening and closing, of ice age and thaw. A fossil is, in a very real sense, one of the most durable objects of biology produces, and it produces them almost by accident as an extremely rare byproduct of the normal process of death and burial. Now, running alongside the story of the skeleton's physical persistence is a very different story about the information the skeleton contained, because bone is not just a structural material, it is also a repository of biological information, encoded in the DNA of the cells that built it and maintained it throughout life. Osteocytes, the bone cells embedded within the mineral matrix, contain full copies of the organism's genome, the same genetic information found in every other cell of the body. And unlike the soft tissue cells that decomposed weeks or months ago, these cells are partially protected by the surrounding mineral matrix, which provides some physical barrier against the processes that degrade DNA. But some protection is not the same as full protection, and DNA is a fragile molecule in ways that minerals are not. It is a long chain of nucleotides held together by chemical bonds that are constantly susceptible to damage from multiple directions. Hydrolysis, the breaking of bonds by water, occurs spontaneously and continuously. Oxidative damage from free radicals fragments the chain. Radiation, including background cosmic radiation and radiation from trace radioactive elements in the soil, causes breaks and cross-linking. In a living cell, all of this damage is continuously repaired by a dedicated suite of molecular machinery. After death, the repair stops. The damage accumulates without any corrective mechanism operating on it. The long, continuous strands of DNA begin to break into shorter and shorter fragments over time, like a very long piece of string that is being randomly snipped at thousands of points simultaneously. The rate of degradation depends heavily on environmental conditions. In permafrost, cold, stable, dark, and largely anoxic DNA degradation slows dramatically, and ancient DNA researchers have recovered readable genetic sequences from permafrost specimens approaching 1 million years old, and in rare exceptional cases, somewhat beyond that. From warm, wet burial environments, usable DNA may survive only decades to a few centuries. In most conditions, somewhere between those extremes, DNA in skeletal remains degrades to the point of unrecoverability within tens of thousands of years at most. This creates a striking asymmetry that I find genuinely worth sitting with. The structural matter of the body, the calcium and phosphate of the bones, and in rare cases the mineral replica produced by fossilization, can persist for millions or hundreds of millions of years. The informational content of the body, the genetic code that specified this particular organism with this particular genome, this particular set of inherited characteristics, is gone within thousands of years at the outside, and usually much sooner. The body's structure, in a sense, enormously outlives its identity. The physical scaffolding persists. The instructions that built the scaffolding, and the instructions that distinguish this scaffolding from every other scaffolding that has ever existed, dissolve relatively quickly into the chemical background. There is something quietly striking about that. The bones persist. The genome does not. The shape of the person can be preserved in mineral form for geological ages. But what made that person genetically unique, the specific sequence of 3 billion base pairs that no other organism in the history of life has ever carried in exactly that combination, is gone within a time frame that is short even by human historical standards. Structure outlives information. Form outlives identity. The fossil, if one forms, preserves the container long after everything that specified what went inside the container has dispersed. And even the bones eventually follow. In poor preservation environments, skeletal remains fragment and dissolve within decades or centuries. The hydroxyapatite dissolves into the soil solution. The fragments are physically broken down by freeze-thaw cycles, by root penetration, by the mechanical action of water and wind. The calcium and phosphate ions are taken up by soil organisms, incorporated into plant roots, cycled through the food web. The mineral content of the skeleton re-enters the same biogeochemical cycles that have been circulating calcium and phosphorus through the biosphere for billions of years. Even the most durable part of the body, the part that survives longest, eventually gives way and rejoins the larger flow. The cascade, which has been running since the first 30 seconds, is still running. It has just shifted to a timescale measured in centuries and millennia rather than minutes and hours, and it is not finished. Not even close. Part 10. The ecological redistribution of you. While the bones are slowly weathering through their centuries-long dissolution, something else has already been happening. Something that actually began within days of death and has been running in parallel with everything else the entire time. The soft tissues decomposed. The gases escaped. The fluids leached into the soil. And embedded in all of that, carried along by every chemical reaction and every microbial meal and every molecule of carbon dioxide exhaled by every bacterium feeding on what used to be a person, the atoms have been leaving, not disappearing, not being destroyed, entering the larger systems of the planet, rejoining cycles that were running long before the person was born and will keep running long after every physical trace of them is gone. This is the stage of the cascade where the scale changes most dramatically, where the story of one body opens outward into the story of the entire planet, and where something that might sound like poetry turns out to be straightforward biogeochemistry. Let's start with carbon, because carbon is the atom that defines organic chemistry and therefore defines life, and because the carbon story is one of the most well-mapped elemental cycles on Earth. Your body, by dry mass, is roughly half carbon. It is in every protein, every fat, every nucleic acid, every structural molecule that makes up your tissues. That carbon came from food, which came from plants or animals that ate plants, and the plants got it by pulling carbon dioxide out of the atmosphere through photosynthesis and fixing it into organic molecules. You spent your life cycling dietary carbon through your body, using some of it to build structures, burning some of it for energy, and exhaling it as carbon dioxide, excreting some of it in various waste forms. At death, whatever carbon remained in your tissues enters decomposition, and from there it takes several simultaneous paths outward. Some of it is consumed by the bacteria doing the work of putrefaction, incorporated into their cell walls and membranes and metabolic machinery. Those bacteria are consumed by other organisms, protozoa, nematodes, soil arthropods, and the carbon moves up the soil food web. Some of it is released directly as carbon dioxide, the metabolic exhaust of bacterial respiration, and enters the atmosphere within days to weeks of decomposition beginning. Under anaerobic conditions in waterlogged soils, in deep burial environments, some of the carbon is released as methane instead, a more potent greenhouse gas that eventually oxidizes to carbon dioxide in the upper atmosphere over timescales of decades. Some of the carbon, particularly from more resistant organic compounds, binds to soil mineral particles and persists in the soil as organic matter for years to centuries before eventually being mineralized. But here is the thing about atmospheric carbon dioxide. It does not stay in the atmosphere long by planetary standards. Plants are pulling it out continuously through photosynthesis, converting it back into organic molecules, building it into leaves and wood and roots. The average carbon dioxide molecule spends only a few years in the atmosphere before being incorporated into a plant somewhere, which means that the carbon dioxide exhaled by the bacteria eating your tissues will, within a few years, be pulled out of the atmosphere by some plant, quite possibly on a different continent from where your body decomposed and built into a leaf, or a root, or a piece of wood. From there, it might be eaten by an insect, or a deer, or a person. It might be burned in a forest fire and returned to the atmosphere. It might fall to the forest floor as leaf litter and be decomposed by fungi. The carbon that was in you is now in the global carbon cycle, indistinguishable from the carbon that came from a fallen tree in Borneo or a dead whale on the floor of the Pacific. It is just carbon now. It belongs to the planet. Nitrogen follows a similar trajectory, but with its own characteristic chemistry. Your body contains nitrogen primarily in proteins, the amino acids that make up every enzyme, every structural protein, every antibody, every molecule of muscle, and in nucleic acids, the nitrogen-containing bases of DNA and RNA. That nitrogen was originally fixed from the atmosphere by nitrogen-fixing bacteria, incorporated into soil compounds. taken up by plants, eaten by animals, concentrated and recycled through food webs before eventually ending up in you. When decomposition releases it from your tissues, it re-enters the soil nitrogen cycle, informs that soil bacteria can process ammonium ions, which nitrifying bacteria convert to nitrates, which plants can absorb directly through their roots. The nitrogen from your proteins becomes fertilizer for whatever plants happen to be growing in the vicinity of your decomposition. It enters new plant tissue. It is eaten by herbivores. It passes through food webs. Within years to a few decades, it has been distributed so widely through the ecosystems surrounding your burial site and beyond, through the movement of animals and the flow of water, that tracing any specific nitrogen atom back to you becomes essentially impossible. It has rejoined the communal pool. Water deserves special attention because water is the largest single component of your body by mass. You are, at the moment of death, somewhere around 60% water by weight. That is a lot of water, tens of litres, depending on body size, and it does not stay where it is for long. Some of it evaporates directly from the body surface during the early post-mortem period. Some of it is released by decomposing tissues and drains into the soil. Some of it is metabolically produced by the bacteria consuming your tissues and is exhaled as water vapor or released into the soil solution. It percolates through the soil column, joins the groundwater, moves through the hydrological system toward rivers and eventually the ocean. Or it evaporates from the soil surface and enters the atmosphere directly, where it may travel thousands of kilometers before precipitating somewhere entirely different. Water cycles through the planet's various reservoirs: atmosphere, rivers, groundwater, ocean, ice on timescales that range from weeks for atmospheric water vapor to thousands of years for deep groundwater and glacial ice. The water that was in your body will, on average, reach the ocean within years and cycle back through the atmosphere within decades. After that, it could fall as rain on a mountain range in Central Asia, or be drunk by a child in Brazil, or be incorporated into a glacier in Greenland that will hold it for 10,000 years before releasing it to the sea. The water does not remember where it came from. It is just water now. It belongs to the hydrological cycle, which belongs to the planet. The trace elements follow their own characteristic paths, each one governed by the specific chemistry of that element and the particular biogeochemical cycle it participates in. Iron, which your body concentrated in hemoglobin to carry oxygen through your blood, is released into the soil during decomposition and becomes part of the soil iron chemistry. It may be taken up by plants, where it functions as a cofactor in photosynthetic enzymes. It may be incorporated into soil minerals. It moves slowly through the iron cycle, which operates on timescales of decades to centuries in soils, longer in marine sediments. Phosphorus, which was in your DNA and in the ATP that powered every cellular process throughout your life, is one of the most biologically important elements in the cycle and one of the most actively recycled. Phosphorus moves from soil to plant to animal to decomposer and back to soil in a relatively tight local cycle, with some loss to waterways and eventually to marine sediments, where it can be locked up for millions of years before being released by geological uplift. Calcium, potassium, magnesium, sulfur, zinc, selenium, iodine, each element has its own cycle, its own characteristic residence times in different reservoirs, its own pathway through the living and non-living systems of the planet. And here is the framing I want you to hold, because I think it is one of the more quietly profound things you can say about what a living body actually is. Every atom in your body right now was part of the planet long before it was part of you. The carbon was in the atmosphere as carbon dioxide, or in the soil as organic matter, or in another organism's tissues, before photosynthesis and the food chain concentrated it into the molecules that ended up in your cells. The nitrogen was in the soil or the atmosphere. The water was in the ocean or a river or a cloud. The iron was in the soil. None of these atoms are native to you. They are borrowed. You are, in the most literal chemical sense, a temporary configuration of planetary material, a pattern that the Earth's atoms have been arranged into for the duration of your life, and from which they will disperse back into the planet's cycles when that arrangement ends. Death, from this perspective, is not the destruction of anything. Not a single atom is lost. Not a single proton disappears. What ends is the arrangement, the specific, improbable, energetically maintained configuration that constituted you as a living organism. The atoms that participated in that arrangement are released back into circulation, available to be borrowed by the next organism that needs them. A tree, a bacterium, a person not yet born. The planet runs a closed loop. Almost no material arrives from space in quantities that matter on human timescales, and almost nothing escapes into space. The atoms cycle. They have been cycling for 4.5 billion years, passing through uncountable organisms, uncountable configurations, uncountable arrangements that each briefly called themselves alive before dissolving and returning the atoms to the pool. You are one such arrangement, one temporary borrowing from a pool that has been circulating since before there was life to borrow from it. And when the borrowing ends, the pool gets its atoms back. It always does. It always has. The cycle, as far as we can tell, does not have an end, at least not on any timescale that has meaning for anything living on this planet today. But even the planetary cycle is not the final scale of this story, because the atoms in that cycle, the atoms currently in your body, did not originate on Earth. And where they actually came from is where the cascade opens out into something that makes everything we have discussed so far look like a very small and very local prologue. Part 11. The Stellar Origin Story. If you keep zooming out, and I want you to keep zooming out, because this is where the cascade becomes something genuinely hard to hold in your head in the best possible way, the timeline keeps expanding. Every scale we have visited so far in this story has been larger than the one before it. Minutes gave way to hours. Hours gave way to days and weeks. Weeks gave way to centuries and millennia of skeletal persistence. Millennia gave way to the planetary biogeochemical cycles that redistribute atoms across continents and oceans on timescales of years to millions of years. And now we zoom out one more time, past the planet entirely, past the solar system, past the history of Earth itself, to the question of where the atoms actually came from in the first place. Because they did not originate here, not a single one of them. Every atom in your body arrived on Earth from somewhere else, forged in processes so violent and so ancient that the entire history of life on this planet is a footnote by comparison. Let's start with the simplest atom, because it has the oldest story. Hydrogen is the most abundant element in the universe, and the most abundant element in your body by atom count. Your body is mostly water, and water is 2 hydrogen atoms bonded to 1 oxygen. The hydrogen atoms in that water, in your blood, in your cells, in the fluid that fills every space between your tissues, are primordial. They were not made in stars. They predate stars entirely. They formed in the first few minutes after the Big Bang, around 13.8 billion years ago, when the universe had cooled enough from its initial state for protons and electrons to combine into neutral hydrogen atoms. That process, called recombination, took a few 100,000 years to complete, and when it did, the universe was filled almost entirely with hydrogen and a smaller amount of helium, with trace quantities of lithium. That was essentially everything. No carbon, no oxygen, no nitrogen, no iron, just hydrogen, helium, and a universe full of time. Those primordial hydrogen atoms have been around for nearly the entire age of the universe. They have had extraordinary careers. Many of them were pulled by gravity into the first stars, where they were fused into helium in stellar cores. Some survived in the outer layers of stars and were expelled back into space when those stars died. Some drifted through interstellar space for billions of years in cold molecular clouds. Some were incorporated into planets. The hydrogen atoms currently in the water of your tissues have each had their own individual history across cosmological time, a history of which your body represents the most recent and one of the briefest chapters. Those atoms are 13.8 billion years old. Your relationship with them has lasted, at most, a few decades. The asymmetry is almost funny if you let yourself think about it. Carbon is a younger story, but a more dramatic one. Carbon does not form in the Big Bang. The Big Bang produces hydrogen, helium, and tiny amounts of lithium, and that is where its nucleosynthesis stops. Carbon requires stars, and specifically, it requires a very particular and rather improbable nuclear reaction that takes place inside red giant stars. When a star like our Sun exhausts the hydrogen in its core, it begins to fuse helium instead. Helium fusion produces beryllium-8, an unstable isotope that normally decays back into 2 helium nuclei almost instantly within about 10 to the power of minus 16 seconds, which is a very short time by any standard. But in the dense, hot core of a red giant, occasionally, a third helium nucleus collides with the beryllium-8 before it can decay, producing carbon-1,2. This reaction, called the triple alpha process, is the only significant source of carbon in the universe. And it only works because of a remarkable coincidence. Carbon-1,2 happens to have an energy level, called a resonance state, that makes the triple alpha reaction far more likely than it would otherwise be. The physicist Fred Hoyle predicted this resonance in 1953, specifically because carbon exists and life exists, and something had to have made the carbon. He worked backward from the existence of life to the nuclear physics that must have enabled it, and he was right. The resonance was confirmed experimentally shortly afterward. Every carbon atom in your body exists because a nuclear resonance in carbon-1,2 happens to have exactly the right energy to make stellar carbon production efficient. Change that number, and the universe makes almost no carbon, and there is no organic chemistry, and there is no life. The carbon made in red giant cores is scattered into space when those stars shed their outer layers at the end of their lives, producing beautiful, expanding shells of gas and dust called planetary nebulae, one of the more misleading names in astronomy, since they have nothing to do with planets, but they are genuinely beautiful objects. The carbon drifts into the interstellar medium, where it mixes with other stellar ejecta, where it can be incorporated into new molecular clouds, where it can eventually participate in the formation of new stars and the rocky planets around them. The carbon atoms in your DNA, in your proteins, in every organic molecule that makes you biologically functional, were forged in the cores of stars that died billions of years before the Sun formed. They drifted through interstellar space, were swept up in the collapse of the molecular cloud that became our solar system about 4.5 billion years ago, were incorporated into the rocky material of the early Earth, were cycled through the geosphere and the early ocean, and eventually through the first living cells, and have been moving through the biosphere ever since, through uncountable organisms across billions of years until they arrived, temporarily, in you. Oxygen follows a similar story to carbon, also produced in stellar fusion processes, both in red giants through further helium burning and in the more massive stars that go on to produce heavier elements. Nitrogen, which is in every amino acid and every nucleotide in your body, is also primarily a product of stellar nucleosynthesis, produced in various stages of stellar evolution, and distributed by stellar winds and supernovae. These elements, the big four of organic chemistry, hydrogen, carbon, nitrogen, and oxygen between them, account for over 96% of your body's mass, and their cosmic origins span from the Big Bang to the interior of stars that lived and died billions of years before Earth existed. The heavier elements have more violent origins. Iron is the one I want to dwell on for a moment. Because it is such a central element in human biology, it is what makes your blood red. It is the atom at the center of the hemoglobin molecule that carries oxygen from your lungs to every cell in your body, and its origin is correspondingly dramatic. Iron is not made in ordinary stars like the Sun. It is made in the cores of massive stars, stars many times more massive than the Sun during the final stages of their lives. A massive star spends millions of years fusing progressively heavier elements in its core: hydrogen to helium, helium to carbon, carbon to neon, neon to oxygen, oxygen to silicon, each stage releasing less energy than the one before, until finally the core is converting silicon to iron, and iron is where fusion stops producing energy. Fusing iron absorbs energy rather than releasing it. When a massive star accumulates an iron core large enough, the energy source that has been holding the core up against gravity for millions of years suddenly fails. The core collapses in a fraction of a second. The outer layers of the star crash inward and then rebound in a catastrophic explosion, a core collapse supernova that briefly outshines the entire galaxy it belongs to. The iron from the core is dispersed into space by the explosion, along with a range of other heavy elements synthesized in the extreme conditions of the supernova itself. The iron in your hemoglobin, in your enzymes, in every iron-dependent biological process in your body, was made in the core of a massive star and scattered into space by its death. And then there are the heaviest elements, the ones beyond iron on the periodic table, the ones that require conditions even more extreme than a supernova to produce. For decades, astrophysicists debated the precise origin of elements like gold, platinum, and the rare heavy elements found in trace quantities throughout the body. The leading candidate was neutron star mergers events, where two neutron stars, the incredibly dense remnants left behind by supernova explosions, spiral together over billions of years and finally collide. In 2017, gravitational wave detectors picked up the signal of exactly such a merger for the first time, and the optical follow-up observations confirmed what the models had predicted. The merger was producing heavy elements in quantities consistent with being a major source of gold, platinum, and other R-process elements in the universe. The gold in a wedding ring, if you are wearing one, was almost certainly forged in the collision of two neutron stars somewhere in the Milky Way before the solar system existed, and distributed into the interstellar medium, where it eventually became part of the cloud that collapsed to form the Sun and its planets, and, eventually, you. So the atoms in your body have a cosmic biography that makes your biological lifespan look like a rounding error. Hydrogen, from the first minutes of the universe, 13.8 billion years old. Carbon and oxygen from red giants that lived and died before the Sun formed. Iron from a supernova, scattered into space by an explosion of almost incomprehensible violence. Gold and other heavy elements from the collision of two neutron stars, an event that briefly released more energy than the Sun will produce in its entire 10 billion year lifetime. All of these atoms drifted through interstellar space for billions of years, were gathered by gravity into the molecular cloud that became our solar system, settled into the rocky body of the early Earth, were cycled through geology and ocean chemistry and the deep history of life, and were borrowed, temporarily, by you. You are, in a sense that is not poetic, but strictly chemical and nuclear, a temporary arrangement of material that is almost as old as time. The cascade we have been following from the 30-second gamma surge to the planetary cycles to this has no beginning that belongs to you, and no ending that belongs to you either. Your atoms were in this cascade long before you existed. They will be in it long after every trace of you is gone. The Sun will eventually exhaust its fuel and expand into a red giant, swallowing the inner planets and scattering the atoms of everything that ever lived on Earth back into the interstellar medium. Those atoms will drift, as they have drifted before, until gravity gathers them into new clouds, new stars, new planets, new configurations. Some of those configurations, in some distant galaxy, in some remote future that no human concept of time adequately captures may briefly call themselves alive, and in them, somewhere, will be atoms that were once in you, still going, still moving, still part of the cascade that has no end. Part 12. The final question science cannot close. So let me try to bring all of this together. We have covered a lot of ground tonight, more ground in terms of actual physical distance and timescale than most videos attempt. And I think the picture, when you hold it whole in your mind for a moment, is worth the effort of holding. Because the cascade we have walked through is not just a sequence of biological events. It is one of the most complete descriptions science has ever assembled of what a physical system does when it transitions from one state to another. And the system in question is a human being. So it deserves to be seen clearly, in full, before we get to the question I have been building toward since the first minute of this video. Let me recap the layers, because I want you to feel the scale of what we have mapped. It begins in the first 30 seconds. The heart has stopped. Blood flow to the brain has ceased. By every clinical definition we use, the person is dying. And yet the brain, deprived of its blood supply, deprived of the oxygen and glucose that power every neuron, does something that no straightforward model of brain function would predict. It surges. Gamma wave activity, the high-frequency oscillations associated in healthy, waking brains with conscious perception, attention, and the binding of information across brain regions, increases in amplitude and coherence. Cross-frequency coupling, the interaction between gamma waves and slower rhythms like theta and alpha, which in living people is associated with memory retrieval and complex cognitive integration, also intensifies. By some measures, the dying brain, in those first 30 seconds, is producing more organized, more synchronized, more coherent electrical activity than it produced at any point during the life that just ended. We can measure this. We have measured it in both animal models and human patients in intensive care. The data is real. Then, by the second minute, the energy crisis bites in earnest. The sodium-potassium pumps that maintain the electrical gradients across neural membranes, the gradients without which neurons cannot signal, run out of ATP and stop. A wave of electrochemical failure, called spreading depolarization, begins to move across the cortex at a few millimeters per minute, which is unimaginably slow by neural standards, but unstoppable. Behind the wave, neurons lose their membrane potential. The tissue collapses into silence. The flat line that appears on the EEG monitor is not the moment of death, it is the aftermath, the record of what the wave left behind. Within minutes to hours, the biochemical demolition begins. Calcium floods into neurons through receptors that can no longer be regulated. Proteases and lipases and nucleases are activated by the calcium overload and begin dismantling the cellular architecture from within. Synapses, those extraordinarily precise molecular structures that stored everything learned and remembered across a lifetime, the physical substrate of personality and memory and identity, begin to disintegrate. The brain is not simply losing power, it is being actively taken apart by its own machinery. Machinery that was always present and always capable of this, held in check throughout life by the constant energetic effort to suppress it. Hours after death, the muscles begin to lock. The ATP reserves that allowed actin and myosin to cycle through contraction and release are depleted, and without ATP, myosin cannot detach from actin. The cross bridges lock. The body stiffens in rigor mortis, progressing from the small muscles to the large ones over the course of hours, reaching peak rigidity around 12 hours after death, and then slowly releasing not because anything has recovered, but because the proteins themselves are being digested by autolytic enzymes, and the lock dissolves as the lock itself falls apart. Within hours to days, the microbial takeover begins. The immune system, which spent the entire life of the organism containing the 30 to 40 trillion bacterial cells that lived within it, shuts down as its cells run out of energy. The barriers fall. The bacteria in the gut begin to spread into surrounding tissues. Putrefaction begins the bacterial consumption of the body, producing cadaverine and putrescine and hydrogen sulfide, and a cascade of other chemical byproducts, producing gas that distends the body cavities, producing the chemical signatures that attract insects from distances that, given the body's new lack of opinions about anything, it can no longer object to. Blowflies arrive and lay eggs. Larvae hatch. A predictable succession of arthropods follows, each species arriving at its characteristic stage, each one consuming the substrate and giving way to the next, in a sequence so consistent that forensic entomologists can read it like a clock. Over weeks to months, autolysis and putrefaction work together to liquefy the soft tissues. The carefully maintained chemical compartments of the living body, the dozens of distinct environments separated by membranes and barriers, each maintained at its own precise pH and ionic composition and molecular constituency, merge into a single chemical medium. Organs lose their structure and identity. The face, which in life was the primary surface of recognition and connection, becomes a chemical surface and nothing more. Fluids leach into the surrounding soil, altering its chemistry in ways that persist for years, feeding the plants and microbes in the immediate vicinity with a pulse of nutrients derived from the dissolution of a person. What remains, after months to years, is the skeleton. And the skeleton operates on a completely different clock. Bone is a composite of hydroxyapatite mineral and collagen protein, and while the collagen degrades over decades to centuries, the mineral matrix can persist for thousands of years under favorable conditions, and can fossilize, be replaced atom by atom with minerals from the surrounding rock into a structure that survives for hundreds of millions of years. The DNA inside the bone cells degrades much faster, fragmenting from hydrolysis and oxidation and radiation damage over decades to thousands of years at most, until the genetic information that specified this particular organism, this particular genome, is gone entirely. The structure outlives the identity. The bones may persist in recognizable form long after the instructions that built them have dissolved into the chemical background. Eventually, even the bones go. The hydroxyapatite dissolves into the soil solution. The mineral content enters the calcium cycle, taken up by plant roots, passed through food webs, slowly dispersed. Within years, for most of the body's easily mobilized atoms, within centuries to millennia, for the more persistent mineral components, the matter that made up the person has re-entered the biogeochemical cycles of the planet. The carbon enters the global carbon cycle, cycling through atmosphere and plants and animals and soil microbes on timescales of years to decades. The nitrogen enters the soil nitrogen cycle, fertilizing plants, passing through food webs, eventually being denitrified back to atmospheric nitrogen by soil bacteria and beginning the cycle again. The water, the largest single component of the body by mass, returns to the hydrological cycle within years, evaporating into the atmosphere, precipitating somewhere entirely different, joining rivers and groundwater and oceans, moving through the planetary water cycle that has been running for 4 billion years. The iron, the phosphorus, the sulfur, the calcium, the trace elements, each one rejoins its own characteristic cycle, following its own pathway through the living and non-living systems of the planet, on its own characteristic timescale. And then the scale expands one final time. The atoms themselves did not originate on Earth. The hydrogen in your water is primordial, forged in the first minutes after the Big Bang, 13.8 billion years old, older than stars, older than galaxies in their current form, older than almost everything in the observable universe except the universe itself. The carbon in your DNA was forged in the cores of red giant stars through the triple alpha process, a nuclear reaction so improbable that it only works because of a resonant state in carbon, 1,2, that Fred Hoyle predicted from first principles in 1953. Because carbon exists and therefore it had to work. The iron in your blood was made in the cores of massive stars in their final moments before core collapse supernovae scattered it into the interstellar medium. The heaviest elements in your body, the trace gold, the platinum, the rare heavy atoms, were forged in the collision of neutron stars, events that briefly outshine galaxies and that we can now detect as gravitational wave signals rippling through the fabric of space-time. All of these atoms drifted through interstellar space for billions of years, were gathered by gravity into the molecular cloud that became our solar system 4.5 billion years ago, became part of the early Earth, were cycled through geology and ocean chemistry and the deep history of life across billions of years of biological evolution, and were borrowed for a brief window by you. After the cascade completes, after everything we have described has run its course, those atoms go on. They rejoin the planetary cycles. They pass through other organisms. They persist in the soil and the ocean and the atmosphere. On the longest timescales, they will leave Earth entirely when the Sun expands into a red giant in about 5 billion years, and the inner planets are consumed, and the atoms of every human being who ever lived are scattered back into interstellar space to begin their next chapter in the cosmic cycle that has no end we can currently imagine. Every step of this is mapped. Every step has data behind it. Every step is the subject of active scientific research, with peer-reviewed literature and accumulated observations and increasingly precise measurements. We know what happens to a body after death in more detail, across more orders of magnitude of time, than we know almost anything else about the physical world. The cascade, from the first 30 seconds to the cosmic dispersal of atoms across deep time, is one of the most completely described processes in all of science. And there is one question that sits at the very beginning of it that all of that description cannot touch. The question is this: what, if anything, is being experienced during that first 30-second window of organized neural activity? Not what is happening physically. We know what is happening physically. The gamma waves, the coherence, the cross-frequency coupling, we can measure all of it, chart it, compare it to waking brain states, analyze it in as much detail as our instruments allow. The physical side of the opening event is, paradoxically, one of the better documented parts of the entire cascade. What we cannot determine, what no physical measurement, however precise, can determine, is whether anything corresponds to those physical events from the inside, whether there is something it is like to be the brain producing that surge, whether anyone is home. This is the hard problem of consciousness, and I want to be precise about what that phrase means, because it is often used loosely. The philosopher David Chalmers coined it in 1995 to describe a specific and genuinely difficult gap in our understanding. The easy problems of consciousness, and Chalmers calls them easy, not because they are simple, but because they are the kind of problems that science knows, in principle, how to solve, are the problems of explaining cognitive functions. How does the brain integrate information from different senses? How does it direct attention? How does it regulate behavior? These are hard scientific questions, but they are questions about mechanisms, and science has a well-developed toolkit for investigating mechanisms. We can study them with brain imaging and lesion studies and computational models, and we can make progress. The hard problem is different in kind. It is the question of why any of those mechanisms are accompanied by subjective experience at all. Why does the integration of sensory information feel like something from the inside? Why is there a subjective quality to seeing red, or hearing a particular piece of music, or remembering a face? Why is there an inside perspective at all? We can describe the neural correlates of consciousness, the brain states that reliably accompany conscious experience in healthy waking subjects. But the correlation between a brain state and an experience does not explain why the brain state is accompanied by an experience. It just tells us that it is. The explanation of why physical processes give rise to subjective experience, or even a satisfying account of what kind of explanation would count as an answer, does not currently exist. And this is not a gap that more data straightforwardly fills, because data describes the physical side, and the hard problem is precisely the gap between the physical side and the experiential side. Now, apply this to the dying brain. The gamma surge looks, by every external measure, like a state that in a healthy waking brain would correspond to intense conscious processing. The neural correlates we associate with consciousness, the specific patterns of activity and synchronization and cross-frequency coupling that accompany aware, processing states in living subjects, are present in the surge, in some respects, more strongly than in the waking baseline. If you were looking at the EEG readout without knowing the context, and you applied the standard correlations, you would predict that something significant was being experienced. The brain looks conscious by the measures we use to assess consciousness from the outside. But the standard correlations were developed from studying people who could report their experiences. The dying brain cannot report. It cannot behave. It cannot give us any of the signals we normally use to infer that something is being experienced. And without those signals, we are left with the purely physical data, which is rich and detailed and genuinely strange, and an unbridgeable gap between that data and any answer to the question of experience. Two interpretations dominate the scientific literature on this, and they lead to conclusions so different that it is difficult to overstate the stakes of choosing between them. The disinhibition model holds that the surge is mechanically explained by the failure of inhibitory interneurons during the acute energy crisis. In the normal brain, much of the cortical activity is actively suppressed by inhibitory cells that prevent runaway excitation. When energy fails, these inhibitory cells may fail preferentially, releasing the excitatory network into a brief burst of uninhibited, high-amplitude activity. Under this model, the surge is not organized processing. It is a spasm, the neural equivalent of an electrical system shorting out as it collapses, producing dramatic readings on a meter that correspond to nothing functional happening inside the system. The dying brain, under this interpretation, experiences nothing, because nothing worth calling experience is being computed. The transient cognitive coherence hypothesis reaches the opposite conclusion. It proposes that the surge represents a genuine window of accelerated cognitive integration, that the dying brain, freed from some of its normal regulatory constraints, briefly achieves an unusual degree of coordination across regions that do not normally communicate so strongly, and that this coordination may support real cognitive processing, memory retrieval, emotional consolidation, something like a rapid review of the life that has just ended. Some proponents connect this hypothesis to near-death experience, reports the life review, the sense of peace, the perception of meaningful order, suggesting that the surge might be the physical substrate of those experiences. But this connection is speculative in ways that are difficult to resolve. The reports come from people who survived, which means the cascade did not complete in them. We are inferring from incomplete cases what happens in complete ones. The logical move is understandable, but it cannot be validated, and it leaves the hypothesis in a genuinely uncertain state. What I want to be honest with you about is that we are not stuck simply because we lack data. We could, in principle, record every neuron in the dying brain at perfect temporal and spatial resolution. We could map every synaptic connection, every chemical change, every electrical event in the most complete detail imaginable. And we would still not have answered the hard problem. The data would describe the physical process completely. It would say nothing about whether the process is accompanied by anything from the inside, because that is not the kind of question that physical data by itself answers. The hard problem is not a problem of measurement precision. It is a structural feature of the difference between describing a system from the outside and knowing what it is like to be that system from the inside. Those are two different kinds of knowing, and we have very well-developed tools for the first kind and essentially no tools for the second. So when I say there is one question science cannot close, I mean something specific and important. I do not mean we have not collected enough data yet. I mean that the methods of empirical science, as we currently understand them, are not equipped to answer questions about subjective experience directly. They can describe correlates. They can map mechanisms. They cannot, on their own, tell us whether a system has an inside perspective, because inside perspectives are not the kind of thing that appears in physical measurements. Other minds, in general, are inferred rather than directly observed. You cannot directly verify that any other person is conscious. You infer it from their behavior and their reports, and your assumption of similarity to yourself. A dying brain that can neither behave nor report leaves us without the inferential tools we normally rely on, facing a gap that no current method bridges. The cascade, then, has this strange double character that I find genuinely worth sitting with. It is fully mapped on the physical level and fundamentally untranslatable at the experiential level. And these two failures of understanding are not symmetrical. The physical mapping is incomplete, only in the sense that science is always incomplete. There is more detail to fill in, more mechanisms to clarify, more timescales to pin down. The experiential gap is incomplete in a different sense. It is not clear that filling in more physical detail would close it at all, because the gap is not between different physical descriptions, but between physical description and something that may be outside the reach of physical description entirely. Death, for science, is two stories told at once. The first story, the physical cascade, from the neural surge to the dispersal of atoms across cosmic time, is one of the most complete and extraordinary stories science has ever assembled, and I hope this video has given you some sense of its scope and its detail and its genuinely strange beauty. The second story, what, if anything, the person experiences in those first 30 seconds, cannot be written from external observations. It may never be writable. It sits at the intersection of neuroscience and philosophy of mind, in a place where neither discipline currently has the tools to resolve it. The closing image I want to leave you with is simple. A brain, somewhere, a few minutes after a heart has stopped. The monitors show gamma waves at amplitudes that exceed anything recorded during waking life. Cross-frequency coupling that, in a healthy brain, would correspond to memory retrieval and complex integration. The full electrical architecture of awareness, in some sense, running at the highest pitch it has ever reached, in a system that will be irreversibly gone within another minute. Whether anything is happening from the inside, whether anyone is experiencing that final surge of organized activity, whether there is something it is like to be that brain in those 30 seconds, we do not know. We can describe the outside with extraordinary precision. We cannot reach the inside at all. The map ends there. Not because we stop mapping, but because what is on the other side of that boundary is not the kind of thing that maps, as we currently make them, can show. The cascade is real. Every step of it is documented. The matter goes back into the cycles. The cycles go back into the cosmos. And at the very beginning of it all, in the window, before any of the physical dispersal begins, there is a brain doing something that science can see but cannot interpret, in the dimension that matters most. Hold close, while you can, the strange and temporary and deeply improbable configuration of cosmic matter that gets to call itself you. The atoms are older than you can imagine and will outlast anything you can plan for. You are the part of them that, for a brief window, gets to wonder what they are doing. That, in itself, is the strangest and most remarkable thing in this entire cascade. And it is happening right now, in you, while you watch this. Until next time.